Hello guys, very warm welcome or welcome back to my channel. I am Sasmita. Today we will learn about plasma membrane and it is part 3 video, and if you haven't watched my plasma membrane part 1 and 2, then go and check out our video, otherwise you will not able to understand this one. So let's get into the video. Before that, please guys do like, share and subscribe to my channel, and don't forget to press the bell icon for notification. So guys, let's start our video. Very first, plasma membrane. Plasma membrane made of with three types of component. One is protein, another one is lipid, and then carbohydrates. Protein and lipid present majority of the plasma membrane, but in case of carbohydrate, it is present only 5 to 10% of the total plasma membrane. We have already know in our previous video that lipid composed of three types of lipid present in the plasma membrane, and one is phospholipid, second one is glycolipid, and third one is sterol. Phospholipid, we have covered this topic in our previous video. Today we will see about glycolipid and sterol. So now we will see glycolipid. Glyco means carbohydrate, lipid means lipid. So glycolipid is a carbohydrate which is covalently linked with the lipid. That means carbohydrate covalently linked with a lipid is known as glycolipid. So, if you will ask me what is carbohydrate, then let me tell you, carbohydrate is called so because their general formula has a ratio of carbon, hydrogen and oxygen is 1 is to 2 is to 1. So, it is appear with carbon, and there is H2O water with it. So, hydrate of carbon, so carbohydrate and general formula is CnH2nOn. Then the next one is lipid plus carbohydrate by glycosidic bond or covalent bond. Like lipid conjugate with carbohydrate linked with carbohydrate by glycosidic bond or covalent bond. Glycosiding or covalent bond is a type of bond which link between lipid and carbohydrate with each other. Then if we will see the if we will talk about the location of glycolipid, then we will see it is mainly found glycolipid are found in the surface of eukaryotic cell membrane, eukaryotic cell membrane, and it is extended from the phospholipid to the outer environment. You can see this over this picture, glycolipid is extended out from the phospholipid. Over this picture you can see enzymes called glycoside transferase linked to the saccharides to the lipid molecules. If we will talk about the enzyme, like as I had already said, lipid combines with carbohydrate and make glycolipid. Very obvious, there must be an enzyme who plays an important role in the synthesis of glycolipid, known as glycoside transferase. It links with the saccharides, like means sugar, to the lipid molecule, sugar with the lipid molecules, and from glycolipids. You can say that lipid plus carbohydrate, lipid with carbohydrate, with the help of glycosidic bond and the help of enzyme glycoside transferase form glycolipid. We will see the types. There are two types. One is sphingoglycolipid and another one is glyceroglycolipid. If we will see the structure of sphingoglycolipid, then we will find sphingosine backbone with one fatty acid and carbohydrate instead of phosphate. Then if we will see the glyceroglycolipid, then you will find glycerol instead of sphingosine with two fatty acid, and again a carbohydrate chain, in this case particularly sulfate in the head group. Then the next point is the essential feature of glycolipid is the presence of monosaccharide bind to the lipid moiety. And the next one is the most complex of the glycolipid, the gangliosides. Contain oligosaccharide with one or two sialic acid moiety, which give rise ganglioside a net negative charge. Like ganglioside, it is an example of glycolipid. It contain oligosaccharide, we call it as sugar, with the with one or two sialic acid, which gives rise ganglioside to negative charge. Because of sialic acid, it gives negative charge. And ganglioside most abundant, ganglioside most abundant in plasma membrane of nerve cell, but also found in other cell types, enterocytes, but most abundant in nerve cell. Then if we will see, the simplest glycolipid called cerebroside contains single sugar residues with either glucose or galactose. Now we will see its function. Cell-cell interaction. It interacts with one cell with another cell, and facilitates cellular recognition, means one cell recognize other cell by glycolipids. Then the next point is, it also help in immune response and blood types too. And then the next point is maintain membrane stability. Like, it is important part of plasma membrane, so it helps in the maintain the stability of plasma membrane. Then the next point is entry point for a certain bacteria, toxin and viruses. Like, both virus and bacteria receptor is ganglioside. Like, you can say glycolipid is a receptor. For example, polyomaviruses and cholera toxin also enter the cell after binding initially to the ganglioside. It enters the cell. Now we will see the third type of lipid, which is sterol. Sterol, also known as steroid alcohol, are the subgroups of the steroid and an important class of organic molecule because it is a type of lipid and it is a subgroup of steroid. Sterol is a subgroup of steroid. Then the next point is the basic structure of steroid containing 4 fused ring, 3 are 6C and 1 is 5. Then the next point is, it occurs naturally in plant, animal and fungi, but in case of plant it contains stigmasterol and cytosterol instead of sterol. Then the next point is, it affects the membrane fluidity. Like when the temperature is high, like when the temperature is warm or the temperature is cold, there is no change in the plasma membrane. Like, temperature will not make it leaky when the temperature is warm. Then now we will see the types of sterol. Like, if we will talk about the types, then we will see different types in different organisms, but function is same. Like, the first one is juicesterol. Juice mean animal, in animal including cholesterol. Like, the cholesterol present in animal, we call it as juicesterol. Then the second one is ergosterol. It is present in the fungi plasma membrane. Then the third point is usually absent in prokaryotes, only phospholipid is present. Like, I had already said, you prokaryotes don't have sterol. So guys, it was our third part of plasma membrane session. Hope you all had a good session. Thank you so much for watching our video and thank you so much for your love and support towards me. Keep loving, keep watching. Thank you so much.